All right, welcome to week 4 of Bio 110. We're going to start our lectures about membrane transport. So this first page, slide that I'm showing you, it's just the objectives that I would like you to master about this material. I'm going to leave them there for you to read on your own time, so we don't have to spend some time with them during the actual lecture. But please take the time to look at them. So now, let's take a quick overview about membrane transport. The transport across cell membranes depends on concentration gradients, on the concentration gradients of ions like potassium, sodium, and calcium, metabolite concentration, like the concentration of sugars, as well as electrical currents. Those 3 points, the concentration gradient as well as electrical currents, are going to be extremely important when we begin to understand the concept of electrochemical gradients. Now, in order to allow some of those molecules to come inside the cell, we need to have transport proteins. And transport proteins are going to affect concentration gradients. They're going to either work with them or against them. So you can have carriers and channels. And if you're moving a solute against a concentration gradient, you have to spend energy. So that energy is always required because you're moving a molecule against its concentration gradient. And energy generation is often coupled to the transport and concentration gradients of the cell. So we're going to take a moment later to understand that if you are moving a molecule against the concentration gradient, you have to use energy, but you can also use the concentration gradient to generate energy. Now, when we look at the membrane, it's going to allow the separation of 2 environments, like we see here on the left. Many molecules are trying to cross the membrane, but more often than not, those molecules are not able to cross the membrane because the permeability of the membrane is extremely limited. In order for some of those molecules to cross the membrane, they require transport proteins, and those are going to allow for the controlled movement of solutes across the membrane. So for example, this receptor here is going to allow for the movement of the circles. This other transport molecule is going to allow for the movement of these triangles from the inside of the cell to the outside, and this other transport molecule is going to allow for the movement of this little rectangles from the outside of the cell to the inside. So now, when we think about the membrane themselves, it's not permeable to a lot of different molecules. It is permeable to gases. It is permeable to oxygen, to nitrogen, for example, and to carbon dioxide. Those are able to diffuse easily across the membrane. So small, nonpolar molecules. Small uncharged polar molecules like ethanol are often able to be permeable across the membrane. But other ones are slightly permeable. That means that sometimes that they will have a little bit difficulty crossing. Water will be one of them, for example. Glycerol will be another one. Now, that is the limit of the permeability of the membrane to allow diffusion. Large uncharged polar molecules, for example sugars like glucose and fructose, are completely impermeable to the membranes because they're polar, and they are not able to pass through the hydrophobic region of the membrane. Those also will include charged polar molecules like amino acids, ATP, glucose who has been phosphorylated, or general proteins or amino acids, completely impermeable. Last but not least, the membrane is impermeable to ions, potassium, magnesium, calcium, etc. They are not capable of passing the membrane. What we see is that in order to get those molecules across, the membrane is going to have transporters. They're going to be transporters in the plasma membrane around the cell that are going to allow nucleotides, sugars, amino acids, or ions to go across. But remember that the cell has a network of organelles which are also membrane bound. To have a different level of environment within the cell, and different within the cytoplasm. So for example, a lysosome. A lysosome is going to have a transporter that is going to allow for the transport of protons, therefore lowering the pH within the lysosome. But so it's going to have different transporters within the mitochondrion. Notice that the outer membrane of the mitochondrion really doesn't have a lot of transporters because the outer membrane of the mitochondrion, it's not a permeability barrier, but the inner membrane is. And therefore, you will have transporters to bring molecules like pyruvate inside the mitochondrial matrix, or transport ATP outside of this mitochondria and ADP inside of the mitochondrion. So depending on the organelle or the membrane that you're looking for, you may find different sets of transporters. Now, small molecules enter the cell via carrier proteins or channel proteins. Here in A, we have an example of a transporter. That transporter is going to undergo conformational changes that are going to allow it to transport a small solute molecule across the membrane. On the other hand, what we have are channel molecules. Channel molecules are going to allow for the great influx of multiple solutes across the channel, for example, in this case, ions, and you're going to have basic difference between the transporters and the ions. We're going to look into this a little bit later, but the channels can have configurations. It could be closed, it could be open, and depending on the ion, that would allow the movement of the molecule either inside of the cell or outside of the cell. Now, we can have carrier proteins, and those carrier proteins may come in three general types. A carrier protein that is only allowed to transfer one kind of molecule across the membrane, it's called a uniport. And uniports are able to either allow a molecule to go inside the cell or outside the cell. But oftentimes we have what is called coupled transport. And coupled transport are transport molecules that are going to allow for the transportation of two molecules, a transported molecule and a co-transported ion, for example. If the transport molecule and the co-transported molecule are going in the same direction, we call that coupled transporter a symport, for simultaneous transport, so symport. However, if one molecule is going inside the cell and the other one is exiting the cell, so their molecules are going in different directions, we call that transporter an antiport, because both molecules are going in different directions within the cell. So again, we have uniports, allowing the movement of only one molecule across the membrane, symporters, which allow the simultaneous transport of two different molecules, or antiporters, which allow the simultaneous transfer of two different molecules but in different directions. Now, what I'm showing in this slide are the different types of transport according to a concentration gradients. On the right, in the figure, I modify it slightly by adding a triangle that is pointing down in the inside from an area. The wider area of the triangle indicates that you have a great concentration of those solutes, in this case in the outside of the cell, and then it points out to the inside where you have a lower concentration of solutes inside the cell. So I have modified the figures with little triangles of this nature to indicate the direction of the gradient. Now, certain molecules, like gases, as we discussed, are able to diffuse across the plasma membrane on their own. So molecules like carbon dioxide, oxygen, nitrogen, they're able to diffuse across the membrane on their own, and they do that by the mechanism of simple diffusion. But we already said that a lot of molecules are impermeable in the membrane, that the membrane is not going to allow them to come through, and therefore they need transporters to facilitate their diffusion. So those include passive transport molecules. And passive transport molecules include channels, or include transporters. Again, if the molecule, for example here the red molecule or the orange molecule, when they are going with their gradient, so going from an area of high concentration to going to an area of low concentration, those molecules are moving along their concentration gradient. Therefore, they will have passive transport through the channel or the transporter, and that it's also considered facilitated diffusion because the proteins facilitate the diffusion of those molecules across the membrane. But here we have also the case of a molecule that is moving against its concentration gradient, the square. The blue square has a lower concentration inside the cytoplasm than in the outside. And therefore, it is going to be for it to be transported outside of the cell, it's going to be moving against its concentration gradient, and that molecule is considered to be actively transported because you require energy to facilitate the transport of that molecule from an area of low concentration to an area of high concentration. So, to conclude, we have molecules that can diffuse on their own, again through the membrane, but we have other molecules which undergo facilitated diffusion because proteins are helping them go across the membrane through passive transport because they're going along their concentration gradient. And then active transport, it's mediated by transporters that spend energy to move molecules against their concentration gradient. Now, carrier proteins facilitate passive transport of specific solutes along their concentration gradients. So here we have, again, in this case it's glucose, shown over here by the blue hexamers. And in this case, the concentration of glucose is greater in the outside than the inside of the cell. So glucose is going to move from a higher concentration of solute outside of the cell to a lower concentration of solute inside the cell. So the glucose transporter, it is a passive transporter that is going to be able to bind glucose, undergo a conformational change, and then be able to release the glucose molecule inside the cytoplasm because the energy because the transporter is using the energy of the glucose gradient to move the molecule from an area of high concentration to low concentration. So the GLUT protein, it will be the glucose transporter protein that is an example of passive transport membrane fusion. Ions pose an interesting issue because different ions have different concentrations across the membrane. For example, highlighted here in the red box are two of the most important ions that we're going to learn about. The first one is sodium. Sodium has a concentration inside the cell of 5 to 15 millimolar. However, that concentration is very different from the extracellular concentration of sodium, which is 145 millimolar. So usually in a cell, the concentration of sodium outside of the cell is much greater than the concentration of sodium inside the cell. On the other hand, potassium, the concentration of potassium inside the cell is about 140 millimolar, which is much greater than the concentration of potassium outside the cell, which is around the 5 millimolar range. So potassium and sodium have gradients across the membrane that are opposed to one another. So those differences in gradient concentration across the membrane lead to the electrochemical gradients across the membrane. And we're going to take a look about this more carefully in a moment. So let's look at the concept of a electrochemical gradient. The electrochemical gradient, it's the result of two factors. As you can see here in A, one factor is going to be the solute concentration gradient across the membrane. In the case of sodium in A, sodium it's a lot more concentrated in the outside, so the concentration of sodium is greater in the outside of the cell than in the inside of the cell. So that is going to have a force to direct sodium from the outside to the inside. But that also generates a membrane potential. And that membrane potential, it's a charge across the membrane, and in this case, the outside of the membrane, it's usually really positively charged, and the inside cytoplasmic region of the membrane, it's very negatively charged. So for a sodium ion to come inside, it is then being influenced by two forces. Number one, the force of the concentration gradient, which is pushing it inside the cell, and the force of the membrane potential, which is allowing those positive ions to move towards the negative cytoplasmic environment. So here, that is illustrated by having a strong green arrow, which is indicating that the sodium ions are going to be driven to move inside of the cell with a lot of force. So the electrochemical gradient, when voltage and concentration gradient work together in the same direction, it's very strong. On the other hand, let's take a look at potassium. The concentration gradient of potassium, it's greater in the inside of the cell than to the outside. So the concentration gradient of potassium will lead the potassium ions to move outside of the cell. But the membrane potential of the membrane potential, it's moving, it's working against those positively charged potassium ions because they really would like to stay close to the negatively charged cytoplasmic region and not move towards the positively charged external region. So in this case, the electrochemical gradient, it's a lot smaller, but still driving the potassium ions to go outside because the chemical gradient, it is working against or in a different direction than the membrane potential. But the net effect of the electrochemical gradient is to allow the potassium ions to move from the inside of the cell to the outside of the cell. So again, when you think about the electrochemical gradient for an ion, you need to take into consideration the membrane potential and the direction of the solute concentration gradient. If they're moving in the same general direction or if they're moving in opposite directions. Now, let's take a look at some forms of active transport. In this case, ion pumps. Ion pumps are going to help move ions across the concentration gradient, so basically against it. Therefore, they require energy. That energy could be provided by ATP or by light. They're always pumps, so you can have a sodium-potassium pump, so a molecule that is going to be able to pump sodium and potassium against the concentration gradients. You can have a calcium pump, or you can have a proton pump. So when we talk again a bit about active transport molecules, we're thinking of proteins that are going to move solutes against their concentration gradient, and therefore they require energy to accomplish that goal. So let's take a look about some types of active transport molecules. We can have coupled pumps. In this case, a coupled pump is a pump that is going to move one molecule against the concentration gradient. Here is the orange molecule that is moving against the concentration gradient, but it's using the energy of the concentration gradient of another molecule to drive that movement. So here, the red squares, their concentration is greater in the outside than in the inside, therefore they're moving with their concentration gradient, and they're helping move the orange square against its electrochemical gradient by using the energy of the red molecule. So again, the concentration gradient of the red molecule, it's going It's favorable from the outside to the inside, and therefore the orange square, it's piggybacking on that energy. The other type of active transport proteins will be an ATP-driven pump, where the solute molecule, here again the orange square, is going to be able to be moved against its electrochemical gradient by using the energy of ATP hydrolysis. Last but not least, we have light-driven pumps, which are going to again be able to move the orange square against its electrochemical gradient by using the energy of light. So again, the yellowish orange square, it's moving against its concentration gradient, and the red molecules, either being the red squares, ATP, or light, are showing the energy source that it's going to be used to power the movement of the yellow molecule. So it could be another molecule concentration gradient, it could be ATP hydrolysis, or it could be light energy. So now, let's take an example about a coupled transporter that is going to use the concentration gradient of one molecule, in this case sodium, to be able to move another molecule, in this case glucose, against its concentration gradient. So shown here in the left, we have the sodium electrochemical gradient. As we discussed, the concentration of sodium is much greater in the outside than it is in the inside of the cell. So the sodium electrochemical gradient drives the movement of sodium molecules to the inside of the cell. And on the right, we have the glucose gradient. And the glucose gradient is indicating of here by the blue triangle, the concentration of glucose molecules. It's much greater inside of the cell than in the outside of the cell. So we're going to move the glucose molecule against its concentration gradient, and the sodium ions are going to move with their concentration gradient. So here we have the symporter. The symporter, it's going to open up to allow the binding of sodium ions. That is going to facilitate the binding of the glucose molecule. So that here in the second image, we have the outwardly open symporter. When both the sodium and glucose molecules are present, the symporter will undergo a conformational change that is going to allow for the release of the glucose molecule as well as the release of the sodium ion. And therefore, after the molecules are released, the symporter will go back into an occluded empty conformation, which will then become open to the outside to repeat the process. So again, the sodium potassium, the glucose sodium symporter is going to use the sodium electrochemical gradient to drive the active importation of glucose against the glucose concentration gradient. As a side note, I would like you to think how does the sodium ions, are going to get back to the outside. So we're going to be looking about that in a little moment, but keep in mind, we are moving sodium ions inside, but the electrochemical gradient of sodium has to be maintained. So eventually we have to pump those sodium ions back to the outside. So think about that point when we look at the next slides. So here, let me show you the first movie at how the intracellular glucose concentration it is used around the epithelium of the lining of the intestine to bring glucose inside the cell and later to be passing glucose into the circulation.
One important task for cells lining the lumen of the gut is the uptake of glucose produced by digestion of food. Yet glucose is typically higher in concentration inside the cells than in the gut, and therefore transporting it into the cell requires energy. To this end, a glucose-sodium symport harvests the energy stored in the sodium gradient to pump glucose into the cell. According to one model, sodium and glucose can both bind to the pump, but the binding of one makes the binding of the other more effective. When the binding sites of the symport are open to the lumen of the gut, the high sodium concentration makes sodium very likely to bind, and thus glucose will bind more efficiently. Because the conformational change of the transporter will only occur when both sodium and glucose binding sites are filled, both solutes are transported across the membrane in strict unison and are released together into the cell. On the cytosolic side of the membrane, the solutes could, in principle, also bind and thus be exported again by the same route that brought them into the cell. However, while there is plenty of glucose inside the cell, there is very little sodium. Therefore, the binding of both types of solutes only occurs very rarely, such that most of the glucose molecules that enter the cell will not leave by the same route. The import is therefore unidirectional.
Sounds like the Space Odyssey movie, the sound. So that movie describes how the glucose sodium symporter is able to bring glucose inside the cell. Let's take a look at what happened next. Before we go there, we're going to look at another pump, and this is the sodium potassium pump, which is an ATP driven ion pump. As we discussed earlier, the electrochemical gradient of sodium will drive the importation of sodium molecules from the outside of the cell to the cytosol. On the other hand, here shown on the right, the potassium electrochemical gradient is going to drive the export of the potassium ions from the cytosol to the extracellular milieu. So how do we then bring out the sodium molecules that, for example, were used in the sodium glucose symporter and export them back to the outside? So that will then take the permeation of the potassium pump, the sodium potassium pump, excuse me. In this case, the sodium potassium pump works in such a way that 3 sodium ions will be pumped with the use of ATP, and simultaneously or concomitantly, you will have the import of 2 potassium ions from the extracellular milieu to the inside of the cell. So by using the energy of ATP hydrolysis, we are able to move the sodium ions against their concentration gradient and the potassium ions against their concentration gradient. One interesting molecule is ubiane, and ubiane, it is a molecule that inhibits potassium binding into the potassium sodium pump, and therefore it could be used to prevent the functioning of the potassium sodium pump. So remember that molecule because we may use it later to try to understand the function of this ATP driven ion pump. So now in the epithelial area of the cell, we have 2 different systems. As we learned earlier, in the epithelium of the lumen of the gut, we have an epithelial layer of cells that are exposed to the gut lumen and also the extracellular fluid in the inside of the basal side of the epithelial layer. Tight junctions are protein complexes that separate the apical side of the cell from the basolateral side of the cell. So then we have an interesting glucose concentration, where the glucose concentration inside the epithelial cell, it's really high, but the glucose concentration in the gut lumen or the extracellular fluid are quite low, and that is indicated here by this arrow, which is blue, dark blue in the middle, and very light blue to white in the edges. Now, in the apical side of the epithelial cell, you have microvilli, which are membrane indentations that go up and down, and those increase the surface area of the epithelial cell, so they will have more area to interact with the gut lumen. In the apical side is where we'll find the sodium-driven glucose symporter, which is going to allow the movement of glucose molecules against their gradient by using the energy of the sodium gradient. So glucose is actively transported from the gut lumen in the inside of the cell. But in the inside of the cell, now we have a different story. Now we have a higher concentration of glucose. And glucose needs to be transported to the extracellular fluid, so then it can be picked up by the blood system and transported across the blood to all the organs. What we have there, it is a passive glucose uniport that is going to allow for the movement of glucose from the inside of the cell to the outside of the extracellular fluid in the basal side, and it's using the concentration of glucose to be able to do that. We also have, it's the sodium potassium pump, which is going to use ATP hydrolysis to allow for the movement of sodium against its concentration gradient, so moving it outside of the cell, and the simultaneous import of potassium against the concentration gradient. Now, one thing that sodium potassium pumps also help is to maintain osmotic balance in animal cells. Shown here in the left, we have the sodium potassium pump, which is helping to bring in the potassium ions, whereas it's exporting the sodium ions and utilizing the energy of ATP hydrolysis. And at that same point, what you have next to it, it's a sodium-driven symporter, like the one that we looked at when we're looking at glucose symported by sodium. So that is forcing the, that is helping the importation of a solute like glucose against its concentration gradient by using the energy in the sodium gradient. So under normal conditions, the concentration of solute in the outside of the cell and the inside of the cell, it's going to be isotonic, meaning that the solute concentrations are equal. And to keep the concentration of the potassium solute equal, we have to have an equal concentration of potassium. So the finesse that is happening in animal cells, it's the balance of the use of potassium to bring in solutes across the membrane with the export of sodium ions using the ATP-driven potassium-sodium pump to keep that solute concentration of potassium equilibrated in the inside of the cell with the sodium concentration outside of the cell. Last is that I'm going to show you is going to be the calcium ion pump. The concentration of calcium, as shown here in the left triangle, inside the cytosol is very low. However, in the endoplasmic reticulum or in the sarcoplasmic reticulum of muscle cells, the concentration of calcium is much greater than the concentration in the cytoplasm. So we always believe that the endoplasmic reticulum in cells or the sarcoplasmic reticulum in muscle, it is a organelle that stores calcium. Now, when calcium is released from the sarcoplasmic reticulum to the cytoplasm, that calcium ions need to be moved again inside the lumen of the sarcoplasmic reticulum against their concentration gradient. So that is where the calcium ion pump comes into play. And that calcium ion pump is able to move 2 calcium ions across the membrane of the sarcoplasmic reticulum when the pump is using the energy of ATP hydrolysis. there is a particular aspartic acid in the cytosolic portion of the pump. You have an activator domain as well as a nucleotide binding domain. The aspartic acid that is going to be phosphorylated, it's found within the nucleotide binding domain of the pump. When calcium binds to the pump, you will have the phosphorylation of the aspartic acid, and the phosphorylation comes from the hydrolysis of ATP. And now that phosphorylated aspartic acid is going to induce a conformational change in the calcium ion pump that is going to allow for the release of calcium into the lumen of the sarcoplasmic reticulum, therefore moving calcium ions across their concentration gradient. Last but not least, I would like to show you the bacteriorhodopsin light-driven pump. In bacteria, the bacteriorhodopsin molecule uses the energy of light to move protons from the cytosol to the extracellular space against the concentration gradient. The concentration gradient of protons is much greater in the outside of the cell than in the inside of the cell, and therefore it requires energy to move protons from the inside of the cell to the outside. Here we have a movie that is going to illustrate this process.
Bacteriorhodopsin is an abundant light-driven proton pump found in the membrane of Halobacter halobium, a purple archaeon that lives in salt marshes in the San Francisco Bay Area. Bacteriorhodopsin is a multi-pass membrane protein that traverses the plasma membrane of the cell with 7 long alpha helices. The helices surround a chromophore, retinal, that is covalently attached to the polypeptide chain and gives the protein and cells their characteristic purple color. Retinal is a long, unsaturated hydrocarbon chain that is covalently attached to a lysine side chain of the protein. When retinal absorbs a photon of light, one of its double bonds isomerizes from a trans to a cis configuration, thus changing the shape of the molecule. The change in retinal's shape causes conformational rearrangements in the surrounding protein. The light-induced isomerization of retinal is the key event in proton pumping. In the excited state, retinal is positioned so that it can transfer a proton to an aspartate side chain, aspartate 85, that is positioned towards the extracellular side of the protein. Aspartate 85 quickly hands off the proton to the extracellular space via a bucket brigade of water molecules. The now negatively charged retinal takes up a proton from another aspartate, aspartate 96. This one is positioned towards the cytosolic face of the protein. Upon reprotonation, the retinal returns to the ground state. Aspartate 96 replenishes its lost proton from the cytosol, and the cycle can repeat. The net result? For each photon absorbed, 1 proton is pumped out of the cell.
So from this movie you can appreciate how the molecule of retinal will undergo a change from trans to cis in one of its double bonds, so it's basically losing a proton, and therefore because of that particular event, it will collaborate with the side chains of the amino acids within the bacteriorhodopsin pump to allow the movement of protons from the cytosolic portion of the cell to the extracellular portion of the cell. Now, let's take a moment to look at the difference or the comparison between ion pumps and ion channels. So ion pumps are going to be molecules that use active transport to move solutes, or ions in this case, across the membrane, but against their concentration gradient. Now, channels are molecules that are protein molecules that are going to allow for the rapid, facilitated movement of specific ions across the membrane, but using their concentration gradients. So, an ion channel, it's a facilitated diffusion kind of molecule, versus an ion pump uses active transport. Now, ions are really interesting because they could be gated, meaning that they're closed and then open only when they receive a signal, or they're constantly open. And the other thing is that where an ion pump, like the sodium-potassium pump, only moves between 2 to 3 ions at a time, a channel can move between 10 to the 7 to 10 to the 8 ions per second through the channel. So a much greater flux of ions across the channel than across the ion pump. Now, when we look at channels, we know that channels are selective molecules. They will only allow for the transport of their specific ion across the membrane. So in the one in the left, we have a closed gated channel, and on the right, we have an open channel, but that open channel has a region which is called a selectivity filter that is only going to allow it to pass the ion that is designed to transport. So for example, potassium. Now, how does the selectivity channel work? So here we have an image of the potassium channel. The potassium ion, shown here as a yellow diamond, it's usually surrounded by water molecules where it is in solution. Remember, the concentration of potassium is much greater in the cytosol than in the outside of the cell. So a potassium ion is going to be surrounded by a lot of water molecules, and when it moves inside the channel, it passes by the selectivity filter area, where only the potassium molecule and not the adjacent water molecules is able to pass. We're going to look at the mechanism of that in a moment. Once it is past the selectivity filter, the dehydrated potassium ions are then able to encounter more water molecules to rehydrate and therefore be able to be transported back into that region of the plasma membrane. So when we look at the non-gated potassium channel, this is a tetramer. So a molecule that has 4 subunits, and each single subunit contains 2 alpha helixes, so an outer helix and an inner helix, and a small here shown, the outer helix and the inner helix are shown here in yellow, and then you have a p segment which has a small alpha helix but then a loop, and that loop is what is going to form the selectivity filter. Now, when you put them all together and you look at them from the top to the bottom, you can see the 4 different molecules and how the selectivity filters in the tetrameric channel narrow the position where the potassium ion can pass. From the side, you can see that the channel will have the vestibule, which is the greater area within the channel, and then the potassium ions have to pass through the selectivity filter, which is composed of the 4 loops, one each provided by each of the single units. Now, the interesting part of that is that in the selectivity loop, you have a series of carbonyl groups that have oxygen molecules. So those oxygen molecules, as you know, are going to have a polar charge that is negative. So when the potassium ion that is surrounded by water in the vestibule passes through, the positively charged potassium ion will be able to be shuffled across the selectivity filter by the negative charge of the carbonyl oxygens found within the selectivity loops. To illustrate this, let me show you this movie that is going to show you how the potassium channel works.
The bacterial potassium channel is a multi-pass transmembrane protein in the plasma membrane. It is built from 4 identical subunits that are arranged symmetrically. A pore in the center of the protein allows selective passage of potassium ions across the membrane. 4 rigid protein loops, one contributed by each subunit, form a selectivity filter at the narrowest part of the pore. This structure is responsible for the channel's high degree of selectivity for potassium ions over sodium ions. In this selectivity filter, carbonyl groups line the walls of the pore. These carbonyl groups are spaced precisely to interact with an unsolvated potassium ion, balancing the energy required to remove its hydration shell. Passage of a sodium ion through the channel is energetically unfavorable because the sodium is too small for optimal interaction with the carbonyl groups.
All right. So as you can see, the selectivity filter area with the carbonyl groups allows the oxygens of the carbonyl groups to interact with potassium, because potassium has a perfect fit, but is not able to interact with sodium because the sodium ion is too small. Now, let's take another look at some gated ion channels and how they're able to respond to different stimuli. One of the stimuli that the gated ion channel responds, it's voltage. As we have discussed, you have a voltage potential, so the membrane potential, across the membrane. So for example, in the upper part here in green, you can see that the extracellular charge is positive and the intracellular charge is negative. But you have then, when the membrane undergoes a depolarization event, the charges in the membrane reverts. So the membrane becomes more negatively in the outside and more positive in the inside. Those changes in voltage are going to force the opening of the channel by inducing a conformational change. An example of that is going to be the voltage-gated sodium or potassium channels that are found in nerve and muscle cells. Once the membrane goes back to its original potential state, therefore having more positive charges in the outside and negative charges in the cytosolic side, the channel will close again. Other channels also work as receptors, and therefore they're able to bind ligand. Some of the ligand binding sites are in the extracellular domain, so that will be, for example, the neurotransmitter channels for acetylcholine. So when the ligand is present and it binds to the channel in the extracellular portion, that will force the channel to undergo a conformational change that is going to open it up. Other ones have the ligand binding domain in the cytosolic side. So when the ligand binds to the domain in the same fashion, it will then force the channel to undergo a conformational change that it will open it up. Last but not least, we have some mechanosensitive channels. Those channels are able to feel the stress induced by mechanical forces to the membrane. As you know, the membrane can expand and move around. So when the channel feels those shear-induced mechanical forces, it will open up, allowing for the now transfer of ions across the membrane. So we call those the stress-gated channels. And we're going to look at them like the ones in the hair cells in the ear. Now, for example, here is the ion channels that are in the hair cells in the ear, and those are going to be stress activated, so they're mechanochannels. As we discussed earlier in lecture, the ear have a set of cells called auditory hair cells. Those auditory hair cells, shown here in green, have a set of cilia called stereocilia. And the stereocilia are bundled together tightly. One of the things that also are present in the stereocilia are channels which are mechanosensitive, and they are tethered by a linking filament to the next filament in the stereocilia, shown here in the part B. So whenever there is a sound wave that goes inside the ear that is going to force the bundle to tilt to an angle, in this case to the left, and that tilting results in the linked fragment from one of the stereocilia to now induce a force on the closed channel, forcing a conformational change that is going to allow now for the entry of ions through the membrane. That then will cause a depolarization of the membrane that is going to eventually be received and understood as a signal by the auditory nerve fibers. So in that way, the mechanical movement of the cells results in the conformational change that will open the channel. So to end the lecture, I would like to show you a table that illustrates the mechanism for transport ions and small molecules against the membrane. So what you have is the mechanism of passive diffusion, or transport mechanisms like facilitated diffusion by proteins, active transport, or co-transport, like secondary active transport. So again, take a moment to look at this table so you can understand and be able to compare and contrast the different forms of movement across the membrane based on the properties of requiring a specific protein, solute transported against its gradient, the energy coupled to ATP hydrolysis, if the movement is driven by co-transported of a different ion down its gradient, and then some of the example of molecules that are being transported. Also, at the end, I would like to show you some resources that you can use. I don't know if this diffusion simulator will work in your computer. It used to work on mine, and I was checking it again, and it didn't do much in my new version with Chrome. But it is a beautiful animation that will allow you to see how molecules are transported against their gradient. Two very informative movies are here in the McGraw-Hill website, one of them showing facilitated diffusion and the other one showing the sodium-potassium pump. And last but not least, look at the diffusion versus passive transport information at the biologyabout.com website. With that, we'll finish this lecture, and hope that you're having a good week, and come prepared with questions for Wednesday.